that wonderful presentation. We all learned a lot, and I will also discuss with you offline couple of things. So our next speaker is Dr. Nimesh, and Dr. Nimesh is is leading the Vaccine Immunology Laboratory at National Institute of Immunology. He is a virologist and immunologist by training, and he has obtained his doctoral degree from Defense Research and Development Establishment. He did his postdoctoral training in immunology at Institut Pasteur and University Pierre and Marie Curie, France. He has received several awards from prominent international and national societies for his significant and novel contributions in the field of T-cell immunology. He has also contributed to several high-impact publications in the prominent journals including Science Translational Medicine, Nature Microbiology, Nature Communication, and many more. He has over 25 publications and 2 patents, and he is also serving as an expert in national and international task forces and advisory board of biopharmaceutical industry. We are very fortunate to have you here, Dr. Nimesh. I welcome you for your talk.
Thank you so much, Subrata, for a nice introduction and a very good morning to all of you. So let me start, by thanking the organizers, particularly Ratnesh, to invite me here. And I must congratulate the team, the whole organizing team, for a very successful and a very productive event. So what will I do here in next 20 minutes or so, that I'll take you away from the initial 2 experts, Dr. Abhishek and Nivedita, about talking about the antibodies. I'll take you to the different components of the immune system on the T cells, and we'll share some of our experience in developing some of the T cell based assays at a very innovation stage, so that we can actually take it up for evaluating not only vaccines but the biotherapeutics like the antibodies. So just to introduce that, in my lab, which we incepted in 2016 in the National Institute of Immunology, we basically work on understanding how we actually have a long-lasting protective immunity. What kind of immunological determinants decide that protection? Can be utilized that understanding then to rationalize the development of vaccines. We have various models which we utilize, including COVID-19, which was of course the need of the hour. And these are the major things that we work upon. We looked into the kind of immune memory that we develop in humans after natural infection or after vaccination. And we also look into some of the T cells which are actually necessary for the durable broad protection. And while we do this, it's human is a very complex, a setup to work with, because if you work with human, you just can't go to the tissues very frequently. You have to work with the blood. You talk about blood cells. You cannot, get more than 5 to 6 ml of the blood to develop any kind of assay. And then you're talking about the antigen specificity. It's rare to rarest, the number of cells that you can see in blood. So that's where it becomes important to actually develop some of those assays which can allow us to not only address your fundamental questions, but can also help the vaccine developers, the biotherapeutic developers, to at least characterize their molecules at a very early stage of the development, which can then give them a lot of confidence. The question comes is, if we talk about T cells, I think almost all of us know that level of immunology where we know T cells are important. But why, how, at what level they actually exist is something which we are recently understanding, right? So this is something which we know, that T cells are necessary to establish the humoral responses. When we talk about humoral, it becomes very important to tell you that these are the cells, some of them which are very specialized subsets, which are necessary for devising the quality, the quantity, and the durability of the antibody. If you see, every 3 of these aspects becomes very important when you develop, other molecules like the vaccines. And of course, they constitute immune memory, they mediate cell killing, they drive immunogenicity. That becomes very important again when you talk about the monoclonal antibodies development, right? Because anyways, you have to address those questions at a very early stages so that you can actually have a lot of a confidence on your molecule that you do not have immunogenicity, and it is not very straightforward and simple aspect. It has to be seen both at the early stages and at the later stages. I'll give you some examples of that how we are now helping some of the pharmas in India to actually look into these aspects. Something which we are actually putting a lot of weight is with these T cells, what you can do is you can actually do vaccine assessment at very early stages. It becomes very important during this pandemic because of two reasons. One, when you come up with the new molecules, new vaccine candidates, you do not have the way to really do the trials like you can do with at a very naive situation, right? In pandemic, most of us got infected, and then we got some double doses of the vaccine from the vaccine that exists. Now, how will you do a new candidate trial in humans? And of course, the regulators will ask you to do something, and that's where this concept of bridging trial came, the immune bridging trial, and that's where the T cell becomes the most important aspect. I'll share some of that information that how we are actually moving and recently helped in couple of pharmas in India to do this trial as well. So we talk about T cells, we know everything about T cells. Why can't we actually have a lot of T cell assays? Do you see it's so frequent? No, you don't see those frequently utilized T cell based assays in any industry, right? The reasons are this. These are the limitations. A specialized setup. You really need a very specialized setup to look into the T cell based endpoints, the T cell based readouts or the parameters. High cost. You can understand that when you talk about a specialized setup, the high cost becomes the second, and I will say even the primary scenario where every industry do not want to invest to that level to develop, to innovate, and then to take it, validate, and then utilize in the development of their biotherapeutics or the vaccines. Because actually you need a lot of investment there, including a lot of risk investment. I think we were hearing about it a lot in yesterday's discussions for the whole day, that risk investment, innovation, everything can't go hand in hand, and that's why you need a lot of collaborations. They are laborious, right? Unlike some of the assays in case of looking into the magnitude of antibodies, the quantity of antibodies, what you can do is that you may have maximum, let's say 3 to 4 hours runtime. Here it can go from a day to a week duration to at least look into not into their quantities, but also into their functions. And then comes the know-how. I think that is also very important aspect, because what we have seen in the last slide, that we have a lot of understanding on the T cells, their biology. But how to take that physiological aspect of those T cells to the test tube, that becomes very difficult, because you cannot 100% recapitulate the aspects of T cells from tissues, from circulation, from blood to the test tube, to the plate. So that's the question, that with these lot of these limitations, still we started learning about the various parameters of T cells. And when we talk about the parameters of T cells, they are those who becomes the readouts of the assays. And that's very important to understand, that what all kind of parameters that you can actually utilize while you want to develop or you want to actually make a T cell based endpoint analysis. One is activation. I think majority of us probably know about the activation aspects, the proliferation of the T cells, the suppression aspects, and these are some of the well-known parameters that have been used in both in the academia and industries while we actually characterize the biotherapeutics or the vaccines, and also some of the drugs. Something which is quite new is the quantification. We have been doing quantification of T cells in case of a lot of vaccine evaluation, but it was actually restricted to the polyclonal quantification of the cell, which was not telling us much about the responses of those vaccines in terms of the T cells. But now we have moved to the next level, which is now we can actually talk about the antigen specific, the vaccine specific, the virus specific T cells in a very few ml of your blood, and which can actually allow us to say whether we are making those responses which are necessary or not. Cytokine based parameters, these are some other functions. If you see this side, these are majority of the functional aspects of T cells that have been utilized. One of them is like the cytokine secretion, most commonly used aspect of the T cell, which you can easily assess by utilizing ELISA or by utilizing the ELISpot-based methods. Something, if you see these two aspects, one is on the B cell health and another on the cytotoxicity, have been very difficult. And a lot of work going on both in academia and industries to see if we can recapitulate these functions. Because if we recapitulate these functions, they could be a very handy tools to actually characterize your molecule even before a phase 1 trial or even at a phase 1 trial, because then you can tweak it accordingly to make it better, to make a formulation better, and before you reach to phase 2 and a larger phase 3, you will have a lot of confidence. So this is the area which is something has been a focus from past 2 decades. If you see these aspects, like in biotherapeutics, this is something has been utilized. Mostly when we talk about ADA, those anti-drug antibody responses, it's very straightforward to look into those kind of assays where you can have dendritic cells and the T cells and see if they're activated, if they're proliferating. That can tell you about the immunogenicity. The suppression assays have been utilized in case of the autoimmune drug trials and as well some of the cell therapies. And as I said, that the cytokine and the quantification has been now the focus for the vaccine evaluations. So something which we have not yet reached to this area, and what will I do now is I will share some of our efforts in this particular area of B cell health, that how we can actually go and take those physiological aspects of the B cell health of the T cells to the test tube level, and how we can utilize those assays to address some of the very key questions. I will stick to the vaccine part for the moment because we have demonstrated the utility of these assays in the vaccine evaluation, but they are still open for majority of the questions that you can address on the antibodies, on the monoclonal antibodies, as well as the polyclonal therapies which are in the field, including IVIG. So before I go directly on and showing you some of the data which we have demonstrated, I'll just give you a small session on why this B cell health is necessary. So if you see this, let's say an example, when you get vaccination, your molecules go inside the nodes, you have an interaction of antigen presenting cells with the T cells, then you have a lot of T cell subsets coming out. You do have an early stage antibody response, which we call a primary response, an extrafollicular response, which is not necessarily to stay in a long term. So something which can come in a long term is this particular interaction, which will lead to a germinal center response, and you have a development of memory, you have a development of long-lived plasma cells secreting antibodies. That's what a vaccine developer needs, not this. But when in early stage you look into antibodies, you can't differentiate antibodies coming from here or coming from here, right? So something which can help at an early stage evaluation is this aspect, this particular crosstalk of a T and a B cell that can actually tell us if you have this function happening, if your vaccine is capable of inducing this function, yes, you will be actually making it, and you can then address it may have a durable humoral immunity. And that will save your 5 years, because after a phase 3 trial, you still wait for a couple of years to talk about memory, whether a vaccine induced memory. And then you talk about whether if induced memory, when to boost it, what kind of schedule should you use. So many of those questions can help, if we can actually look into this particular aspects. So talking about the scenario that all of us, those who are working in the T cell in the domain, this has been an exercise of past 2 decades that we have been trying to develop such as this. So majority of these things which were available starting in 2000, some of the experts in states, they were trying to develop these setups, but it was like very polyclonal, spontaneous, and also super antigen specific setups, which do not give any idea about antigen specificity. And what we found is in these 2 decades, why it does not happen, because of the 2 major reasons. And those 2 major reasons were the survivability of these cells in the cultures, which actually impacts the interaction of these cells, and which actually impacts and negatively influence the output of the assay system. And that's why there was no availability of these particular assays in an antigen-specific setting. When I say antigen-specific setting, it's like an example of SARS coronavirus 2 vaccine. You should know what is happening to the T cells, but not the all T cells exist in your body, the T cells specific to SARS coronavirus 2. And that's where Asghar in lab was working, and he tried to find a way to develop this setup which we can actually address the questions at an antigen-specific level. So of course we reproduce, the things. We reproduce some of these things in the lab about is it really the survival? And yes, we found that the cells do not survive in a particular setup where you do not give them an antigen or you do not give them any kind of stimulation. And then we looked into the presence of antigens. It's the same thing; they don't survive. The something which we then indicated and which we found in the literature is a lot of B cells that who survive in the lymphoma are those B cells which are actually interacting with the myeloid cells in those lymphomas. And that was some hint that we utilized. And we tried with monocyte supplementation in these crosstalks. And what we found is, yes, these cells are capable of actually modifying the survivability of these B cells. So, we did a majority of the assays on understanding the several aspects of control and interactions. And we found that you need to have a good antigen, you need to have the survivability, and if you have a survivability index which is high, along with the presence of antigen, the crosstalk happens and it increases the sensitivity of the assay. I'll just quickly go to the slide which is here, which is on the application of these assays. So we have applied those assays. We have applied those in the SARS coronavirus 2 settings, and we showed that you can actually enhance the reading capacity of the functional aspects of these T cells in COVID, both in infection and vaccination, and that goes up by almost 90 to 100-fold, and which allow to actually look into particularly the sensitivity of these setups. Let me skip a few of them. And here, yeah. So which can actually allow you to look into the sensitivity, which goes down to 90 antigen-specific T cells. And that was so amazing for us because we can read with 90 antigen-specific T cells their function, and then we can tell whether this particular vaccine candidate is capable of inducing those T cells at that level, those who have a good function to establish humoral immunity. So we then applied this setup to the multiple agents by now, variety of vaccines in case of dengue, SARS-CoV-2, CMV, and MTV, and we are continuing to make this assay much more user-friendly so that we can actually apply it much more robustly with the multiple vaccine candidates. So here are some of the key takeaways which I will just comply here, which I already said that this assay is now available, which we are utilizing in the antigen-specific settings. We can use peptides and the whole antigen, and it can read as low as 90 antigen-specific T cells. It's highly specific. It only will work when you have a presence of antigen and a cognate interaction happening between T and B cells. And this method is actually now helping us to evaluate the vaccines in early stages, and we can say whether the vaccine is inducing durable and protective humoral immunity or not. And I think there's more to be done in future to harness the T cell attributes, to develop the assays not only for vaccines, but also for the immunotherapeutics. And this is something which we have developed in our lab, a vaccine evaluation facility. Majority of the aspects we have utilized, and many of these setups are now available, which we can help if anyone is interested from the vaccine manufacturing companies. And this is something which we have shown a real demonstration of this platform. One of that in a post-marketing trial on a COVID-19 vaccine, Covaxin, which we provided a lot of information on the immunological effectiveness of this vaccine. And recently, we also held in Bharat Biotech to evaluate their intranasal COVID-19 vaccine to understand that immune bridging trial, to understand in around 300 donors that whether this vaccine is capable of inducing the mucosal responses or not. And because of this platform, there's a lot of interest from Ministry of Health to generate evidence so that they can make evidence-based policies for the heterologous or the homologous boosters in case of the COVID vaccines. So these are, some of the happy, healthy, and the handsome faces in the lab, those who helped us to reach this level in past 6 years, and many of them have moved to the next stages of their career. And I think this was not possible, what we were able to achieve in this country, without a chain, a good network of our lab with variety of scientists, both from India and outside, and also a lot of network from the clinicians in this country. And of course, I'm really grateful to the faith of these funding agencies, because the working with the human setup is a costly affair, and they really helped us to set up these programs, and now we are helping them back in terms of looking into these aspects of vaccines so that they can actually decide good policies for the Indian population. And with this, I'll stop here. Thank you so much for your time and attention.
Someone, let's say, hypothetically, in a pandemic situation or in a normal, non-pandemic infection, if someone doesn't have an infection, still he receives the vaccine dose, will his memory be less than someone who's received the infection along with the vaccine, or do you need both?
Yeah, that's an interesting question, and let me start saying that the answer to that question is still not available, across the globe. We do not know how infection alone or infection plus vaccination or vaccination alone is actually working and inducing the memory. But I can tell you that as an immunologist, we have seen the hybrid immunity, the infection plus vaccination, is much more efficient in actually imparting not only the long term of the immune memory, but also the quality of immune memory, right? The breadth of the protection that you can see in hybrid immunity is much better. And I think there is a lot of complicated biology exists there. This probably will be achieved if we know. That's what I said, that this is what we are trying to understand. If you can understand that what is that particular aspect, taking you a long-term protection, taking to you a durable memory, taking you to a kind of breadth, a larger breadth of protection against multiple variants. Let's say an example of SARS-CoV-2, it becomes a kind of rich resource for vaccine development, for rational design of vaccines, right? So yes, this is something which is an interesting field. I will say in a decade time, you will have a lot of answers to this particular question.